Hello everyone, welcome to your own YouTube channel Shantanu Bioprep Life Science. Today we are going to discuss electron transport chain or oxidative phosphorylation in detail. In the previous lecture we have discussed glycolysis, Krebs cycle, regulation of Krebs cycle, pyruvate oxidation. In this video we will discuss electron transport chain or oxidative phosphorylation in detail. This topic is given in very complicated way in almost all reference book as well as good sources on internet. So most of the students are worried or scared about this topic. Okay, so you don't need to be worried because we will understand it in very easiest way. Oxidative phosphorylation. Electron transport chain is also known as oxidative phosphorylation. Why? Oxidation means there will be loss of hydrogen atoms and electron, and reduction means acceptance of hydrogen atoms and electrons. So here there will be loss of hydrogen atoms and electrons, and some kind of gradient will be produced of hydrogen ions, and by utilizing that hydrogen ion gradient we will synthesize ATP. That's why the name oxidative phosphorylation. And this one statement we will understand it in upcoming slide in detail. Okay, location of electron transport chain in eukaryotes is in inner mitochondrial membrane because eukaryotes has membrane bound cell organelles. In prokaryote it occurs in plasma membrane. So this can be a favorite question of paper sitters, that in prokaryotes where does the electron transport chain occur. So it will occur in plasma membrane and not in cytosol. Okay, glycolysis occur in cytosol in prokaryotes, but here oxidative phosphorylation will occur in plasma membrane in prokaryotes. Okay, so you need to remember this. Second point is that electrons are transferred from NADH and FADH2 to terminal electron acceptor, that is oxygen, through a series of electron carrier, and that is what is happening in electron transport chain. Okay, there are four complexes in electron transport chain. Complex 1 is known as NADH coenzyme Q reductase, or also known as NADH dehydrogenase. Complex 2 is known as succinate coenzyme Q reductase or succinate dehydrogenase. Complex 3 is coenzyme QH2 cytochrome C reductase or cytochrome BC1 complex. Complex 4 is known as cytochrome C oxidase. Now a question was asked, I think in DBT or ICMR in this year, that which of the following enzyme is present in both Krebs cycle as well as electron transport chain. So the correct answer was succinate dehydrogenase. Okay, so we should remember some important points before we study electron transport chain. The first point is that FeS, that is iron sulfur protein, can accept only one electron at a time. So it can accept only one electron at a time. Mobile electron carriers are ubiquinone and cytochrome C. Mobile means it can diffuse or it can move from one place to another. So it is ubiquinone and cytochrome C. Electron carriers are arranged from lower to higher reduction or redox potential, or from less positive to more positive reduction potential. Okay, so this is a very important point, and a question in DBT JRF 2021 was asked from this. We will understand it. Suppose this is electron carrier A, this is next electron carrier B, and this is terminal electron acceptor oxygen. What is written? Electron carriers in the electron transport chain are arranged from lower to higher reduction or redox potential. Reduction potential means the ability or the potential to accept electron or to accept hydrogen atom. We know that loss of hydrogen atoms and electron is known as oxidation, whereas acceptance of hydrogen atom and hydrogen atom and electron is known as reduction. Reduction potential means the potential or the ability to accept hydrogen atom and electron. So if in the electron transport chain, the electron carriers are arranged from low to high reduction potential or redox potential, it is true statement because electron will be pulled by next electron carrier. Why? In this case, the redox potential or reduction potential of B electron carrier will be higher as compared to A. So if the reduction potential of electron carrier B in this case is higher as compared to A electron carrier, higher reduction potential means higher ability or more ability to get reduced. High ability or more ability or potential to get reduced, which means electron carrier B will have higher potential to get reduced, means it will pull the electron from electron carrier A towards itself. Oxygen will have highest reduction potential in the entire electron transport chain. So as it has highest reduction potential, it means it has highest potential to get reduced, which means it has highest potential to accept electron. If it has highest reduction potential as compared to B electron carrier, it will pull the electron towards itself from electron carrier B. Okay, and that is why the electron carriers are arranged according to lower to higher reduction potential, so that electron will sequentially move and finally get accepted by terminal electron acceptor, that is oxygen. Okay, the question paper sitter can also twist this statement in order to confuse students, that it can be arranged from less positive to more positive reduction potential. Suppose I randomly plug in some values. For example, reduction potential for electron carrier A is 0.50, for B 0.60, and for C, for oxygen 0.70. We know that negatively charged molecule or negatively charged electron will be attracted more by more positively charged thing. We know that. We know this, right? So if we logically think, electron will remain with electron carrier A, or it will get pulled by electron carrier B. In this case, obviously it will be pulled by electron carrier B because this is more positive. So this will strongly attract electron from A towards B because this is more positive. So negatively charged electron will be more attracted toward more positively or more positive thing rather than less positive thing. Yes, that is why electron carriers are arranged from lower to higher reduction or redox potential, or from less positive to more positive reduction potential. Okay. Complex 1 pump 4 hydrogen ions. Complex 2 do not pump any proton. Complex 3 pump 4 proton. Complex 4 pump 2 protons from the matrix to the intermembrane space. Coenzyme Q or ubiquinone exists in 3 redox state. Why is it called Q? Because Q refers to quinone chemical group. Okay. Fully oxidized state known as ubiquinone or COQ or Q, when it accepts one electron or one proton, it will get converted into semi-reduced state, that is semi-ubiquinone or UB semiquinone, represented by this. When semiquinone accept one more electron, it will get converted into fully reduced state, that is ubiquinol or COQH2 or QH2. Or when that is fully oxidized state, when accept 2 electron, it will get converted into fully reduced state, that is ubiquinol. Okay. Reduction means acceptance of proton and electron. So fully oxidized state, after accepting one proton and one electron, it will get reduced into semi-reduced state. Semi-reduced state will again accept one proton and electron, and it will get converted into fully reduced state. So can we say that fully oxidized state after accepting 2 proton and 2 electron will get converted into fully reduced state? Okay, that is ubiquinol. Simple. Coenzyme Q or ubiquinol is the only electron carrier that is not bound to any protein. This can be asked in exam. Iron sulfur proteins. Okay. Let me hide the video so that you can read what is there behind me. Iron sulfur proteins has non-heme ion complex with sulfur. Two common iron sulfur proteins are 2Fe2S and 4Fe4S. Both iron and sulfide ions are coordinated to 4 cysteine sulfidyl groups. So this you need to just memorize it. Okay. You don't need to study this in depth. Okay. Because it is not at all important. There are many things, much information provided in the reference book for this topic, subunits and all. You don't need to remember it. Okay, now we will understand electron transport chain in detail. Let me first draw mitochondria for you. So we know that this is outer membrane. This space is known as intermembrane space. This thing inside is known as matrix, and this is inner membrane space. Sorry, inner mitochondrial membrane, inner membrane. So if we see the expanded view of inner mitochondrial membrane, it will be like this. Okay. So can you imagine? Okay. So this diagram is also very important to understand the basics. Now we will start. NADH molecules are getting produced in glycolysis and Krebs cycle. NADH molecules will enter electron transport chain. It will come and bind to complex 1. This is complex 1. This is complex 3. And this is complex 4. I have purposely not shown complex 2 over here. Okay. Complex 2 we will see later, because complex 2 explanation in this will create much confusion. Okay. And you will understand in the upcoming slide why I have not shown complex 2 over here. Okay. NADH molecule will come and bind with complex 1. It will come and bind with complex 1. Then oxidation of NADH molecule will occur. Oxidation means loss of hydrogen atom and electron will occur. So if there is loss of hydrogen atom there will be two electrons released. How? We know that in the previous lecture we have studied that NAD plus will accept two hydrogen atoms. Out of two hydrogen atoms, one hydrogen atom will be accepted as a hydride ion. This is hydride ion, and one hydrogen ion which is left is accepted as it is. Hydride ion means one proton and two electron. We know that NAD plus will accept two hydrogen atoms. So if these are two hydrogen atoms, one hydrogen atom will have one proton, one electron. Similarly over here, one proton and one electron. Yes. So NAD plus will accept one hydride ion, which means one hydrogen ion and two electron, that is hydride ion. So this H is hydride ion. And what is left? This free hydrogen ion, which will be accepted as it is. That is why we write NADH plus H plus and not NADH2, unlike FADH2. Yes. Okay. So now you can imagine that after the oxidation of NADH molecule, two electrons are released. And two H plus ions are also released. Yes. So two H plus ions will be released in the matrix. Okay. So now we know how the two electrons are coming. How these two electrons are coming from the oxidation of NADH molecule, and how two H plus are getting released in the matrix. These two electrons will be accepted by FMN electron carrier, which will get converted into FMH2, that is reduced state. Then FMNH2 will pass one electron to FES, because we have studied in the last slide that FES can accept only one electron at a time. So FMNH2 has transferred one electron to FES. This electron, it will again pass to next FES. From FES, the electron will go and enter in the ubiquinone pool. Okay, ubiquinone pool is in the nearby vicinity of complex 1. So when this electron will be accepted by ubiquinone, it will get converted to UB semiquinone, that is partially reduced state. We have discussed it here. When one electron is accepted, fully oxidized state will be converted into semi-reduced state, that is ubiquinone will be converted into semiquinone. Okay. Now, second electron from FNH2 will be transferred to the FES, because we know that FMNH2 has accepted two electrons. And when FMNH2 will release both of the electrons, it will again get converted into oxidized state, that is FMN. Okay. So second electron will pass to the FES. Then FES will transfer the second electron to next FES molecule. This second electron will get passed to semiquinone, and this semiquinone will get converted into ubiquinol, that is fully reduced state. We have discussed it over here. The semi-reduced state, after accepting one more electron, it will get converted into fully reduced state, that is ubiquinol or QH2. Okay. Now, QH2 or UQH2 will come out of its pool and it will bind to the QP site which is present in the complex number 3. And this site which is present in complex 3 towards the matrix is known as QN site. Okay. So ubiquinol which has been formed will come out of the pool. It will go and bind to the QP site present in the complex number 3. Okay. Before understanding that, when the electrons were getting transferred to the successive electron carriers, some energy will be generated. This energy will be used in the pumping of hydrogen ions or protons from the matrix to the intermembrane space. Okay. So complex 1 will pump 4 hydrogen ions or protons. Okay. Okay. So from this pool, it will come and bind to the QP site. We know that this is fully reduced state, means it has 2 hydrogen ions or 2 hydrogen atoms and 2 electrons. So it will first release its first electron to FES. FES will transfer the first electron to cytochrome C. Cytochrome C will carry the electron and bind to the complex 4. From complex 4, the electron will be passed to CuA, cytochrome A, CuB, cytochrome A3, and ultimately the electron will enter in the matrix. Okay. Next, the second electron from UQH2 will pass to Bl. L stands for low affinity, H stands for high affinity. It will get passed to Bl followed by BH. Okay. Up till now you have understood it. When the UQH2, that is ubiquinol, lose 2 electron, it will get converted into ubiquinone. Because ubiquinone is the fully oxidized state, it will accept 2 hydrogen atom and 2 electron and get converted into ubiquinol or UQH2. So if UQH2 lose 2 hydrogen atom and 2 electron, it will again get converted into UQ, that is fully oxidized state. Okay. So from UQH2, 2 hydrogen will be released in the intermembrane space. And this first electron and this second electron is getting released, due to which it will be converted into oxidized state, that is ubiquinone or UQ. UQ will again enter pool. So after UQ enter its pool, this QP site will be vacant. This will be now vacant or empty. Okay. From pool, ubiquinone will enter and bind to the QN site which is present in the complex number 3. This UQ or ubiquinone will accept this electron which was getting transferred from here, and ubiquinone will get converted into semi-reduced state, that is UB semiquinone. Okay. Clear till now? And this QP side has now become vacant. We have studied this. Next thing. So as this QP site was vacant, a fresh molecule of or second molecule of UQH2 will come out of the pool again, and it will bind with the QP site. This is QP site in the complex number 3. Again the same story will continue. It will again lose 2 protons and 2 electrons. First electron will pass through FES, second electron will take this route and will get accepted by Bl. After losing 2 protons and 2 electrons, fully reduced UQH2 will get converted into fully oxidized ubiquinone, and ubiquinone will again enter into its pool, which means QP site is again getting vacant or empty. Okay, so up till now 4 H plus or 4 protons is getting released from complex number 3. Okay, so this first electron will be accepted by cytochrome C. Cytochrome C will go and bind to complex 4. It will transfer this electron to CuA, followed by cytochrome A, followed by CuB, followed by cytochrome A3, and finally it will enter matrix. Now previously this electron was also there, and this is second electron. From this second electron is also coming. We know that positive things will be attracted by negative things. So 2 H plus ions will combine with 2 electrons. One hydrogen atom has 1 proton and 1 electron. So can we say that hydrogen atom is getting formed? Yes, 1 proton, 1 electron is hydrogen atom. So this is first hydrogen atom and this is second hydrogen atom. Okay, which means it will 2 H plus or 2 hydrogen will combine with oxygen to form water molecule. Very easy. Okay. Now we will understand what happens at the QN site where the semi-ubiquinone is present. So this semi-quinone was already formed in this, as we can observe. Yes, semi-quinone was formed. This semi-quinone will accept the electron following this pathway or this route, and when the partially or semi-reduced UB semiquinone will accept this electron, it will get converted into fully reduced ubiquinol, right? And this fully reduced ubiquinol, to form this, we will require 2 H plus ions from the matrix. So I have directly shown 2 over here. See, like this. Now you will understand it better. When UQ will accept 2 hydrogen atoms, it will get converted into UQH2. Okay. So for this we require 1 H plus, and for this we require 1 H plus, or to accept 1 H plus. So instead of gradually showing this, and in order to, this will create much confusion. Okay. So instead of showing 1 and 1 H plus ion, I have directly shown 2 H plus ions here. Okay. So UQ or ubiquinone will accept 2 H plus ions from the matrix, and it will get converted into UQH2. Okay. This fully reduced ubiquinol will again enter in the pool, which means QN site is also vacant now or empty. Yes. And we can also observe that from complex number 4, the energy is used to pump 2 H plus ions from the matrix to the intermembrane space. Okay. So this QP site and QN site is now empty. Now the next cycle will again begin. This is pool. From pool, UQH2 will again come and bind with QP site, and story from here will again start. Okay. Right. So 4 plus 4 plus 2, 10 protons are pumped from matrix to intermembrane space when complex 2 is not involved. Okay. Now we will see complex 2. Complex 2, succinate is converted to fumarate, which will be catalyzed by enzyme succinate dehydrogenase. Succinate dehydrogenase has FAD as coenzyme. So when FAD will accept 2 electron, it will get converted into FADH2. So it will now transfer 1 electron at a time to FES, because we know that FES can accept only 1 electron at a time. So this first electron, it will transfer it to the UQ, that is ubiquinone. Then it will get converted into UB semiquinone. Then second electron from FADH2 will transfer to FES, and this second electron, when it will get accepted by UB semiquinone, it will get converted into fully reduced state, that is UQH2, which is ubiquinol. This ubiquinol will come out of the pool and bind to its QP site, which is present at in the complex 3. After this, same story is going to continue. Only the difference is that these 4 H plus ions, which we are going to liberate from complex, which we are going to liberate because of complex 1, will not occur, and the number of protons which will be pumped from the matrix to the intermembrane space when complex 1 is not involved, there will be total liberation of this 4 and this 2, that is total 6 proton when complex 1 is not present. Okay. But complex 2 will not pump any protons from matrix to the intermembrane space because the energy is not sufficient. The protons are getting pumped from complex number 3 and complex number 4. Okay. when complex number 1 is not there. When complex number 1 is also there, then 4 plus 2, we will get 10 protons. Okay. And when complex number 1 is not there, we will get 4 plus 2, that is 6 protons. Okay. Now let us look at the Q cycle summary quickly. Let me hide the video so that you can see what is there behind my video. Okay. First, ubiquinol or UQH2 from pool will come and bind to the QP site which is present on the complex number 3. After that, oxidation of it will occur, which means 2H plus ions will be liberated, will be released to the intermembrane space. 2 electron will be released. After release of 2 hydrogen and 2 electron, fully reduced UQH2 will be converted to fully oxidized UQ, that is ubiquinol. Ubiquinol which has been formed will again enter back to the pool from QP site. Okay. And the 2 electrons which have been released, first electron will be accepted by, or 1 electron will be accepted by cytochrome C, and second electron will be accepted by Q. We know that once the UQ enters back to pool, it will again come and bind to the QN site. So once the QN comes and bind to the QN site, and once it accepts this electron, it will get partially reduced. It will get partially reduced into UB semiquinone at QN site. Yes. And we know that when UQ oxidized, when UQH2 was getting oxidized, after getting oxidized, UQ will enter back to pool, which means this QP site will be empty. We know that. And in QN site there will be UB semiquinone. So this empty QP site, in this empty QP site, second QH2 or second UQH2 from the pool will come out, and it will bind to the QP site, that is vacant QP site or empty QP site, and the same story will continue. It will again get oxidized, releasing 2H plus ions out in the intermembrane space. 1 electron will get accepted by cytochrome C, second electron will get accepted by UB semiquinone, and we know that after accepting 1 electron, UB semiquinone get converted into fully reduced form, that is ubiquinol. Okay. After oxidation of this UQH2 to fully oxidized UQ, this will enter back to pool. Yes, which means again QP site is vacant. And this UB semiquinone, after accepting electron, will get converted into this UQH2. Okay. But we also know that UQ has to accept 2 hydrogen atoms and 2 electron to get reduced to UQH2. We know this. So to completely reduce this, here it will require one H plus ion to get converted into this, and here also it will require one more H plus ion to get converted into this. That is why I have shown directly two H plus ions over here instead of showing 1 plus 1. Okay, to avoid confusion. Okay, so this UQH2, which is now formed at the QN site, will again enter back to its pool, which means there is vacant QN site also now. There is QN site is also vacant now. Now in the next case, what will happen again? Again from pool, UQH2 molecule will again enter and bind with the QP site. And the same story will continue. This was electron transport chain and Q cycle. I hope you have understood this very clearly. Yes. Next. Now next things are very much easier to understand. ATP synthase is also known as complex phi. It is of 6 lakh Dalton. 1 kilodalton has 1000 Daltons. Okay. So 600 kilodalton will be 6 lakh Daltons. It is composed of two components, F0 and F1. ATP synthase is world's smallest molecular motor. F0 component is embedded in inner mitochondrial membrane. Okay. Inner mitochondrial membrane. And has one A subunit, two B subunits, 9 to 12 C subunits. So this is F0 component. It has A, B and C. Okay. C subunit consists of alpha helices which span inner mitochondrial membrane. F0 component is inhibited by oligomycin, therefore we call it F0 or FO. F1 component consists of 3 alpha, 3 beta, gamma, delta and epsilon as shown over here. Only this B will be in F0 component. Okay. F1 component is tightly bound to F0 component. F1 component protrudes towards matrix. Beta subunit is the site of ATP synthesis. This question has been asked in Maharashtra Set Life Science and many other exams. Moving unit rotor, this is important, is gamma epsilon and C9 to 12 subunit ring, whereas stationary unit stator is A subunit, 2 B subunits, alpha, 3 subunits of alpha, 3 subunits of beta and delta complex. When proton pass, moving unit or rotor will rotate in counterclockwise rotation direction. A question isn't asked till now from here, so it can be asked in which direction it will rotate, counterclockwise or clockwise. Okay, so you need to remember 7 point also. Two proton half channel are present in A subunit, which are essential for the movement of protons across the membrane. So A subunit will have two proton half channel, which is very much essential for the movement of protons across the membrane. So these points you have to remember. Okay. Majorly a question which is asked is that F0 component is located where? Okay. So F0 component will be here. So I will draw like this, and this will be F0 and this will be F1 component. Okay, in inner membrane. F1 is in matrix, it is protruding in matrix, and this above portion is intermembrane space. In intermembrane space, we have studied that the protons will be pumped from matrix to intermembrane space, due to which there will be gradient produced, or the concentration of protons in the intermembrane space will be higher. Due to this, along the concentration gradient, the protons will again come to the matrix through ATP synthase. So the protons will be again transferred to the matrix via ATP synthase. When these protons are getting transferred from the intermembrane space to the matrix, this will result in the production of ATP or ATP synthesis. Okay. This proton transport is along the concentration gradient. Why? Along means higher concentration to lower concentration. Movement of molecules from higher concentration to lower concentration. So here higher concentration is in intermembrane space, lower concentration is in matrix. So the movement of molecule will be from higher concentration, that is intermembrane space, to lower concentration, that is matrix. That is why it will move along the concentration gradient. Okay. The question is typically asked, F0 component is present in intermembrane space or matrix or inner membrane space, membrane. F1 component is facing towards the intermembrane space or towards the matrix. Like this, the question will be asked. So you need to remember this diagram. Okay. And which part is stationary and which part is moving unit or rotor? The simple way to understand this is this C ring and this gamma epsilon ring, only this portion takes part in rotation when the H plus ions from the matrix will enter A subunit. Sorry, from this is intermembrane space. Okay. Here the orientation is shown different. Okay. This H plus ion will be released in the matrix, and this is in intermembrane space. Okay. So from intermembrane space, the proton will come and bind with the A subunit, due to which rotation of C ring will occur, and also gamma and epsilon ring will occur. So it is moving unit, and all rest of the things, A, B2, alpha, 3 subunits of alpha, 3 subunits of beta and delta, are stationary unit. They will not participate in rotation. Okay. Rotary motor or engine driving model or binding chain model is a widely accepted model for ATP synthesis. When proton passes through C ring, rotation of gamma epsilon subunit occurs by 120 degree, as a result of which conformational change occurs in 3 beta subunits which drive ATP synthesis. Rotation of gamma subunit, epsilon subunit in response to passing of protons occurs by unknown mechanism. The proton from intermembrane space, when it is going to pass through the A subunit of F0 component, there will be rotation of C ring, and along with it the gamma epsilon subunit will also rotate. But how this rotation occurs is still unknown. Okay. But due to this rotation, there will be change in the conformation in beta subunit. And when one proton passes, 120 degree rotation will occur. Okay. Beta subunits have 3 conformational chain states as follows. O state, that is open state, it binds ATP, ADP and inorganic phosphate very weakly. Loose state or L state binds ADP and inorganic phosphate loosely. T state is tight state, it binds ADP and inorganic phosphate very tightly and it gives ATP. ATP released in the O conformation is energy dependent, so you need to remember this. Beta subunit changes in order OLTO, which we will now understand with the help of diagram. Complete 360 degree rotation of gamma epsilon subunit will release 3 ATPs. This we will also understand with the help of diagram now. These points are important. And beta subunit is the site of ATP synthesis. That point is also important. So you need to study like this. Okay, you don't need to study in much depth, and such information will not be asked in the exam. Okay, so you need to be very smart in picking up those points which are very much important and from where the question can be asked. Okay, now we will understand this 4th and 5th point in detail. In this, to understand these diagrams, you need to first imagine the diagram. Okay, you need to imagine that this diagram, starting from this diagram, it will rotate in 120 degree rotation in the counterclockwise manner. Clockwise means we know clock 12, 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12. So instead of moving like this, we need to move in opposite direction. That is opposite direction of clockwise direction. Okay. So if this is L, here T and O. L means ADP and inorganic phosphate will bind loosely. T means ATP will bind tightly. And in open state, ATP will be released. So in 120 degree rotation, one ATP is going to first ATP release. Now if we rotate it in the 120 degrees in counterclockwise rotation or in counterclockwise way, this L will be over here, T will shift to right side, and O will go up. Yes. Now previously there was ADP and IP, but this L, a loose state, has changed the conformation due to rotation and it has changed to T state. Similarly over here, O state has been changed to L state and T state has been changed to O state due to counterclockwise rotation, because we have seen that due to rotation there is conformational change in beta subunit. Okay. Previously there was ADP plus inorganic phosphate at this side. So when the L conformation will change to T conformation, we know that in T conformation ADP and inorganic phosphate will be tightly associated, due to which we will get ATP. Okay. In the L state, ADP and inorganic phosphate will come and bind weakly. In O state, it will release. So previously there was ATP in tight state, so it was tightly holding it. But when due to rotation, the T state will get converted to O state, there will be release of this ATP. This ATP will be released. So due to 120-degree rotation, this first ATP is getting released. Okay. Now next 120-degree rotation. Okay. So you can again imagine the second diagram. This second diagram to rotate 120-degree counterclockwise. So if we rotate, T will be downwards, O will be towards the left side, and this L, which is present here in downwards, it will go up. Okay. Now previously in the place of T, ATP was there. Due to rotation, this T, the conformation of this T state has been changed to O, due to which this ATP will get released. So this is second ATP which is getting released. In 240-degree rotation, this is second ATP which is getting released. Previously there was ADP and inorganic phosphate at L state conformation. Due to rotation, it is getting converted to T conformation. In T conformation, this ADP and inorganic phosphate will be tightly associated, due to which ATP will form. And L state which is left, we know that ADP and inorganic phosphate will come and bind to it weakly. Now this, after the next 120-degree rotation, we will get first diagram. As easy as that. Yes, it is as simple as that. Okay. This is after 120-degree rotation. You can also imagine it from here, from third figure, that after another 120-degree counterclockwise rotation, O side will be facing downwards, L side will move to left side, and T side will be present above. So here in the third diagram, ADP and IP was weakly bound with the L state. But now this L state is getting converted to T state. And we know that in T state, ADP and IP or inorganic phosphate is held together tightly, due to which ATP synthesis will occur. So we can observe ATP synthesis. Previously there was ADP and IP at L state. Okay. So I have shown T state upper. So this ADP will again come and bind with the loose side, and the ATP which is there in this third diagram, when the T conformation will change to O conformation, in O conformation ATP will be released. So this is third ATP which is getting released. This is third ATP release. Okay, this is third ATP. So can we observe that complete 360-degree rotation of gamma epsilon subunit will release 3 ATPs, as well as 3 ATPs are also getting formed. Okay. And we can also observe one important thing, that the beta subunit conformation changes in order OLTO. See, this O is first, then this L, then this is third, and then again O. Order is OLTO. So a question can be asked in the exam. Okay, you need to remember this. Easy. Next, 4 H plus ions produce 1 ATP through complex 5 or F0 F1 synthetase or ATP synthase, and 10 H plus ion will produce 2.5 ATP, which means when 4 H plus ions let me draw so that you will understand it much better. So instead of drawing that, I will draw like this. Okay, this is F0 F1, and here gamma epsilon complex will be there. Here intermembrane space, this is inner membrane, and this is matrix. We know that the proton gradient is higher in intermembrane space. When four protons will enter through the ATP synthase back into the matrix, we will get 1 ATP. So 4 protons, when getting passed through the ATP synthase from the intermembrane space to the matrix, we will get 1 ATP. So 10 H plus ions which are getting pumped where these 4 and these 2. 10 H plus ions which we have already studied. 10 H plus ions will result in how many ATP? 4x equal to 10, x equal to 10 by 4, that is 2.5 ATP. So 10 protons will produce 2.5 ATP. 1 NADH plus H plus will produce 3 ATPs, and we know that to produce 1 ATP, 4 H plus ions are required. So to produce 3 ATP, 12 H plus ions are required. 12 H plus ion will be required. 1 FADH2 molecule will produce 2 ATP. 1 ATP requires 10 protons to pass from ATP synthase, to pass through ATP synthase. To produce 2 ATP will require 4 into 2, that is 8 protons from through the ATP synthase. Okay. Inhibitors of electron transport chain, this you need to memorize. There is very less or no concept involved to understand this. Okay. Rotenone is also used as a fish poison and as an insecticide. It is derived from plant amityl or amobarbital barbiturates. Pyricidin A, this is antibiotic. It will inhibit complex 1. Malonate carboxyl inhibit complex 2. Antimycin A is also antibiotic. Dimercaprol also inhibit complex 3. Cyanide, azide, carbon monoxide, hydrogen sulfide inhibit complex 4. Oligomycin is also an antibiotic. Remember, it inhibits F0 component of ATP synthase. Dicyclohexyl carbonamide DCCD inhibit complex 5 or ATP synthase. Stigmatilin inhibit electron flow from COQH2 to FES. Plants as alternative rotenone insensitive dehydrogenase, which means even if rotenone is present, it will not result in much, it will not affect much. Okay. And also alternate oxidase. Alternate oxidase is resistant to cyanide. So even if cyanide is present, plants can still survive because it is alternate oxidase and it is resistant to cyanide. But it can be inhibited by salicyl hydroxamide acid. So you need to remember this. Stigmatellin inhibit electron flow from ubiquinol or coenzyme COQH2 to FES. COQH2 to FES. Okay, we will understand it from here. COQH2 to FES. So this route will be inhibited by this electron flow will not occur when stigmatellin will be present. The stigmatellin is inhibitor and it is preventing the transfer of electron through this route. Okay. Next, uncouplers. We know that oxidative phosphorylation or electron transport chain is tightly coupled with phosphorylation or ATP synthesis. Electron transport chain is tightly coupled with ATP synthesis because in electron transport chain, protons will be pumped from matrix to intermembrane space, and using these proton gradient or electrochemical gradient which have been produced or generated in the intermembrane space, the protons can move along the concentration gradient through the ATP synthesis complex back to the matrix, due to which ATP synthesis is going to occur. But uncouplers will separate these processes. Which process? Uncoupler will separate electron transport chain from ATP synthesis. So it will de-link electron transport chain and ATP synthesis process. It will uncouple it or de-link. So oxidation will occur, but ATP synthesis will not occur. Okay. Process by increasing the permeability of inner mitochondrial membrane to protons. This we will study with the help of diagram in some time. It promotes oxidation of substrate like NADH and FADH2 at increased rate but without ATP formation. It will promote oxidation, but it will inhibit ATP synthesis. Examples of uncouplers are 2,4-dinitrophenol, dicumarol, carbonyl cyanide, trifluoromethoxyphenylhydrazone, FCCP, dinitrocresol, pentachlorophenol. FCCP is 100 times more efficient than DNP, okay, to behave as an uncoupler. Aspirin at high concentration, thyroxine at high concentration, thermogenin, valinomycin, gramicidin A and nigericin, etc. Oligomycin can inhibit both oxidation and as well as phosphorylation. Thermogenin is present in brown adipose tissue in upper back and neck, whose role is to produce body heat, especially in newborn babies and also during hibernation in animals like snake. So let us first understand what is this statement. De-link by increasing the permeability of inner mitochondrial membrane to protons. So this is C ring, which is present in F0 component of ATP synthase. This is gamma epsilon complex, and this is F1 subunit, which means alpha, beta, and delta subunit will be there. F1 component. Here it will be again intermembrane space. This is inner mitochondrial membrane, and this is matrix. Okay. Okay. So what uncouplers will do? It will increase the permeability of the inner mitochondrial membrane, which means this membrane for protons. And we know that in intermembrane space there are protons in higher amount. Okay, we know this. Okay, so instead of the protons, instead of passing through ATP synthase, will start to pass through inner membrane space. inner mitochondrial membrane. Why? Because uncouplers have increased the permeability of inner mitochondrial membrane to protons. The protons will now pass through inner mitochondrial membrane rather than passing through ATP synthase. So if the protons will not pass through ATP synthase, will the ATP synthesis occur? It will not occur, because when four protons pass through ATP synthase, it will produce one ATP. But if protons are passing through inner mitochondrial membrane, because the permeability of inner mitochondrial membrane for protons is increased by uncouplers, and the protons are not getting passed through ATP synthase, the ATP synthesis will not occur. And this is how the uncouplers is separating electron transport chain from ATP synthesis. Okay, so there is D-link in the electron transport chain and ATP synthesis process. This is the role of uncouplers. Okay, so ATP synthesis will not occur, but oxidation will continue. Okay, and it also has physiological role for, like, thermogenin is present in brown adipose tissue in upper back and neck, whose role is to produce body heat, especially in newborn babies. In newborn babies, there should be maintenance of temperature. So in order to protect them from cold, this thermogenin can play very important role. It will result in increased oxidation of substrate. So if there is increased oxidation of substrate, obviously heat will be released, because in the last lecture we have discussed that one molecule of glucose when oxidized will produce huge amount of heat in calorimeter. Okay, we have discussed it. It is produced in kilojoules. Okay, so if there is increased oxidation, means more heat, and more heat will protect the newborn babies from cold environment or cold temperature. Okay, it can protect them. Okay, and this thermogenin is also present in animals which hibernate. Okay, so it can also help during hibernation in animals like snakes. And this 2,4 dinitrophenol was previously used for fat loss and weight loss. Okay, because of increased oxidation, more heat will be generated, but ATP synthesis will not occur. So due to increased oxidation, there will be more and more glucose will be oxidized, more and more heat will be liberated. Okay, so it was originally used as in patients who want to reduce fat or weight, but now it has been discontinued because of some side effects. Okay, like increased body temperature, hyperthermia, etc. Okay, so this was about electron transport chain in detail. So I think this video was very much helpful for you, especially for all those students who were actually afraid of this topic. They were saying that this topic is much complicated and they were not able to understand this from any reference book. And actually I have read many reference books, and I have also understood that this was given in very complicated manner. So I have simplified it for you. So I hope it was very much helpful for you. So please don't forget to like, share and subscribe my YouTube channel. And in the next video, we will probably solve the previous year good quality questions from CSIR NET on glycolysis, Krebs cycle, pyruvate oxidation and electron transport chain. Okay. So please don't forget to like, share and subscribe my YouTube channel. And see you in the next video. Thanks for watching and keep practicing. Okay.